Microbiology.
Bioremediation, biotechnology, and biogenesis.
Is biogenesis like where you take DNA and you kind of mix it and you can make things how you want them?
No, but good opinion. Oh, what's genesis?
What?
Creation.
The theory that living cells only rise from free living cells. Biotechnology, I always think of things that help us as technology advancing humankind. So microbes that help in bioremediation. It's really stupid, but I always think of like the mediation between bullies. You have to have someone who's a neutral party in there who will clean it up. I don't know. So I always remember it. It's for the oil spills and stuff. So you're using a microbe to clean up something. Hi. Plug in.
How about 50 for history?
He loves Lily Pasteur. I can almost guarantee you're going to have one question on there. I am. At least one.
Pasteurization.
Yay! Good job. What else?
Oh, the swan flask.
Okay, good job. What else?
What was her name? No. Sorry, we're discussing.
This is.
Good job. You said vaccines for chicken cholera. Remember the lazy assistant?
Oh.
What else?
Oh, and didn't he disprove the spontaneous regeneration thingy?
Very nice. Very nice.
Because I guess that was with the swan flask, but still.
But still, yeah. The one we forgot was that he found the protozoans in the silkworms. So people started to believe that there was actually things that made you sick. Good job. All right. So, I believe we started with Logan.
Oh yeah, it's like your turn.
Yep. So it's your turn. So Logan is 150. And you are 100. By the way, Moa won last night. You and Logan tied.
Whoop whoop. Microbiology.
What are the major categories of microorganisms, and why are microorganisms useful?
They're found all over the body. They help in the digestive tract. They help in the respiratory tract. They decompose. And then the nitrogen cycle and major categories. Archaea. Bacteria. Viruses aren't.
Are they not part of it?
They're not part of eukaryotes and prokaryotes, but yeah.
Archaea is not really part of it, I don't believe, but it would be nice to know. Because
Archaea is like a bacteria.
Yeah, exactly. And archaea are what? Are they eukaryote, prokaryote? Good job. Another way, all of those were right, but you also have to remember making cheese and beer, because he talked a lot about that right at the very first. Good job. Ow, Logan.
Oh, is it big?
No, I'll be fine in a minute.
All right, how about definition for 40?
This is so easy, you can do this.
Solute goes into the water, the solvent.
Is the stuff in the solution that is being dissolved?
Remember the ute goes in the water.
Yeah. Sorry, I didn't know if you could hear her or not. The mic's kind of a little bit away from her.
So an example would be like the solute is the salt and the solvent is the water.
Very nice.
That's
Good job. Thank you. Okay. We'll bring him.
History, 40.
Yeah, she does. She did after. could do that.
The Jenner. Edward Jenner?
Yep.
Very nice. Very nice. What did he use, by the way? What was it called?
Cowpox? Is that what you're talking about?
Yeah, but what was the process called?
Microscope. Oh, yeah.
What?
I just say Scott. I did.
Sure. We'll do microscope for 40.
Cross-reactive immunity?
Okay, what? Staining or microscope?
Microscope.
It reduces the refractive index.
Amplifies magnification. Very nice. All right, Brigham. That's the last 40.
Yes. Right? To see how to treat bacteria. And obviously, you don't want to treat it for the wrong thing, or it's not going to do anything besides get rid of.
And more specifically, like, how would you treat a gram-positive bacteria?
How would we?
Yeah.
Penicillin, right?
Yeah. Yeah.
What does
Something
penicillin
avoid.
do?
What?
Oh,
Yeah.
very nice. Good job. Okay, Logan, your turn.
How about microbe world for 30?
Ocular lens times objective lens.
What?
Ocular lens times objective lens.
Very nice.
Oh, they do it.
That's the only one that I've done
Why didn't it do it?
What are you trying to do? Oh,
You
didn't
did
go to the
30,
thing?
right? It didn't do it. Here we go. Okay,
Oh.
you're getting microbial world, okay? Both one.
Change of where animals are, where they're living.
Okay,
Quicker transportation.
very nice. Population, that's a good one. Girls?
Oh, yeah. It's the girls' fault.
It's always a girl's fault.
Air travel, environmental changes, social changes, the greater population, like you said, evolutionary changes, mass production of tainted food.
Yeah, he might have you list a couple of those.
Yeah, I
Yeah.
remember
He-
doing that one
yeah.
specifically.
So
Okay.
I would remember those.
Okay, Logan.
All right, definition for 30.
Negative, simple, and differential stains.
Negative stains the background, simple stains the microbe directly, and differential. I'm trying to remember differential. Could that be like the sound one where you? Oh, never mind. Okay, I'm saying yes.
Oh, there's the ones in particular. The differential. Okay.
Good job.
Like where you dye it twice and the microbe will take up one specific stain over the other stain.
Did you hear that,
basically.
Logan?
Nope.
Can you hear Brigham at all?
Not really. I can hear you loud and clear.
I'm sorry. Say it again. Can you hear her now?
Can you hear me?
A little bit, yeah.
I was just going into a little bit more detail about the differential stain, saying that you use two different stains, and depending on what kind of bacteria it is, it'll uptake one stain over the other stain. So, just a little more clarification.
Okay, good to know.
That was not what I wanted. Wow. We'll do that one. right. Do you want me to just choose for you?
Yes, please. Well, positive is like the blue, purplish, whatever you want to call it. Negative would be red. And the steps. Well, I just have to remember the order. I can remember like some of them, but.
I use an acronym.
Yeah, what was it? The CIAS, right?
Yeah.
Oh, yeah. CIAS.
Very nice.
Iodine,
Which is the mordant.
and then saccharine.
Yeah, saccharin. You go, girl. Nice.
Wait, what's the acronym?
Yeah, thanks for coming, guys.
Can you write the acronym on the board real quick?
No? CIAs.
CIAs.
Is that for the gram negative one? Oh, okay, that's
Just for
That's
gram
gram.
staining.
It's
Yeah,
not for the
gram
acid
staining.
fast one.
No.
Is there another one for acid fast? The other one for that?
I think.
Acid fast is shorter.
Oh.
Did they?
Aha, there we go. Bring 'em.
Oh, it's not shorter.
Is that the carbon fusion
Schwam.
red one? Is it carbon fusion? And then the heat, and then is that supposed to be wash? And the acid alcohol and methanol fluid.
Schwam. Yep. Okay, so what color is negative? Very nice.
For acid fast?
Acid fast? No. Negative is blue, sorry. Red is the saffron for gram.
Oh, yeah.
Gram. So red for acid fast is opposite, so it's positive. So it's blue is negative, and red is positive.
And then it's the opposite for gram staining.
Yeah.
And if it's, that's like the test they use for tuberculosis, right?
Yes,
Right, and what
exactly.
else?
One more.
Is it leprosy?
Leprosy, very good. Anyone?
Heat or chemical, right?
Okay, very good. Why?
So that. How's it coping by? So you can look at it better.
Very nice. Anyone?
Yeah, didn't we do this one? It was the ocular.
Good job.
Active.
Here we go. Let's try this one. What was John Needham's mistake?
Oh, the hole? It's open.
Yeah, he cross contamination.
Yeah, is he the meat guy? The red meat? I think.
No. He was not the meat guy.
No, I keep thinking. I always get him mixed up with him.
Remember Reddy?
Reddy,
Like
red meat, yeah.
Oh, yeah.
Maybe this, yeah.
Isn't there a John?
Is that
Needham-
like a
Oh.
contamination?
Like the letting in air to his specimen or whatever, because he would say that it was spontaneous regeneration, but it really wasn't because he was letting air in.
Very nice.
Or never mind.
No,
Transfer
it was. It was. This is what the flask kind of disproved. And who was he fighting against? It was Lorenzo's Ballings and something.
Yeah, the weird Italian name.
Yeah, some dead dude. All right.
Some dead dude.
Define resolution and refraction index. resolution is the ability of the lens of the microscope to distinguish between 2 points. So you're trying to figure out if it looks nice, pretty much. Trying to figure out if you can see it. Refraction index is the velocity at which light passes through the specimen. So if you put oil into it, the refractive index is changed so you can see closer. Does that make sense? Kind of?
Mhm.
Thanks, Bobby. Dun dun dun.
It's like the aggressive congregation of microbes.
Very nice.
I don't know why I think like the grouping of it.
That's right.
I don't know why I said congregation.
Yeah, as long as you understand, we're good. What's a prokaryote and a eukaryote? What are the differences? What are the microorganisms under these categories? Oh, Brigham, you.
Prokaryotes have no bound nucleus or whatever, membrane. Eukaryotes do. And prokaryotes are like bacteria, the archaea.
Very nice. So what's under eukaryotes?
Like fungi, protozoan. What else? What else?
Oh. An algae. That was kind of hard. Okay. We already did that one. We already did that one. Why are you not doing this? Oh, okay. What's the size of most bacteria?
0.05 micrometers. Oh.
So if it sees, if a microscope can see below 0.2 UN, then it would be seeing what? Viruses. Very good. We already did that one.
Keeps coming up.
I know. I don't know what's going on. All right. Yay, we're done. And okay. The thing I'm going to do really quick is just ask you a bunch of questions about the first. It was the first 4 chapters, wasn't it? Or no, it was the first 2, 3. Okay, blank is the first name and typically a large group. Blank is the first name and typically called a large group.
Genus?
Genus, very good. What bacterium is commonly found on a human skin? Staphylococcus aureus, very good. Robert Hooke developed a crude multiple lens microscope and examined a cork, which led to the blank theory. Cell theory. Okay. Edward Jennings finds the cowpox can immune a person against smallpox by using blank immunity.
Like cross-reactive.
Thank you. Cross-reactive immunity. Okay. Randolph Virchow claimed living cells only arise from pre-living or pre-existing living cells. This theory is called
Biogenesis.
biogenesis. Nice. Okay. Let's see. What set of rules is used to provide a particular microbe causes a particular disease?
Koch's postulates?
Very good. Okay, this is an example of recombinant DNA technology. This is an example of recombinant DNA technology. It's insulin. Do you remember that?
You can make it from pigs or something like that.
You can take it from a different animal and slice it, just like you were talking about. Okay, let's see. Microbes that break down and recycle what 3 elements usually? What are the most common elements that they like to recycle?
Yeah, perfect.
Nitrogen, oxygen, and carbon.
Very nice. 80% of oxygen comes from trees and forests. True or false?
False.
Why?
Algae.
Yay! I don't know if he'd have this. I remember him having this on one of the tests, but it was that Candidius. It's the symbol of the snake. You know, the two snakes. The symbol for most medical
Oh, mm-hmm.
things. Anyway, the question is true or false? Candidius was fashioned after a worm. True or false?
Wasn't it a snake? No. Probably wasn't.
Is it true?
It's true, actually.
Oh,
I remember he had that.
Yeah.
Yeah.
It was,
That was kind of a
I
random
can't
one.
remember, but yeah, it was like some helmet or something. Something nasty.
Here, I'm gonna put that one. Aha! Here we go. Okay. What is monotrichonous? Monotrichonous.
That's where it has one.
Oh yeah, one flotilla. Big one.
Flagella. Very good. Okay. What if it has flagella all over? What is it called?
Flotilla.
Peri. Very good. I heard it.
Harry. Yeah.
Peri means it's all over. It's all over the body. Peritrichomonas or whatever. Flagella on just one side. Yeah.
Something with L.
Yeah, it's lopo because it's lopsided, remember?
Lopo. That's lopsided.
Let's see.
Can't be.
Multiple flagella on both ends.
Happy.
What?
Ampy.
Uh-huh. Let's see. Transfer of DNA to help with resistance for the bacteria. This helps transfer DNA from one bacteria to another to help with resistance. It's what it's called. It's not the process. What that little thing is called.
Sex highlight?
Sex pili. Very good.
It transfers.
This makes the bacteria go in kind of a corkscrew motion.
Spirochete.
Yep, spirochete.
How do you remember that?
Okay, let's see. True or false? Dead negative bacteria does not cause endotoxic shock.
It's false.
Why?
Is it? Because the capsules still? Or like they can still release toxins.
They can still hurt you. So it is false,
Yeah.
you're right. Okay. That one's too easy. Too easy. Oh, true or false? Human cells can't get hurt by lipozyme.
By what? Sorry?
Lysozyme. True or false? Human cells cannot get hurt by lysozyme. You know, like that's in the tears and then the mucous membrane.
So we don't have peptidoglycan?
Good job, so it would be true. Human cells can't get hurt. This is an example of recombinant DNA technology. It's insulin. Do you remember that?
You can make it from pigs or something like that?
You can take it from a different animal and slice it, just like you were talking about. Okay, let's see. Microbes that break down and recycle what 3 elements usually? What are the most common elements that they like to recycle?
Yeah, perfect.
Nitrogen, oxygen, and carbon.
Very nice. 80% of oxygen comes from trees and forests. True or false?
False.
Why?
Algae.
Yay! I don't know if he'd have this. I remember him having this on one of the tests, but it was that Candidius. It's the symbol of the snake. You know, the two snakes. The symbol for most medical
Oh, mm-hmm.
things. Anyway, the question is true or false? Candidus was fashioned after a worm. True or false?
Wasn't it a snake? No. Probably wasn't.
Is it true?
It's true, actually.
It's
I remember
true.
you had that.
Yeah.
Yeah.
It was,
That was kind of a
I
random
can't
one.
remember, but yeah, it was like some helmet or something. Something nasty.
Here, I'm gonna put that one. Aha! Here we go. Okay. What is monotrichonus? Monotrichonus.
That's where it has one.
Oh yeah, one flotilla.
One flagella. Very good. Okay. What if it has flagella all over? What is it called?
Flagella.
Hairy.
Hairy. Very good. I heard it.
Harry, yeah.
Hairy means it's all over. It's all over the body. Hairy trichomonas or whatever. Flagella on just one side. Yeah.
Something with L.
Yeah, it's lopo because it's lopsided, remember?
Lopo. That's lopsided.
Let's see.
Ampy.
Multiple flagella on both ends.
Ampy.
What?
Ampy.
Uh-huh. Let's see. Transfer of DNA to help with resistance for the bacteria. This helps transfer DNA from one bacteria to another to help with resistance. It's what it's called. It's not the process. What that little thing is called.
Sex highlight?
Sex highlight. Very good.
It transfers.
This makes the bacteria go in kind of a corkscrew motion.
Spirochete.
Yep, spirochete.
How do you remember that?
Okay, let's see. True or false? Dead negative bacteria does not cause endotoxic shock.
It's false.
Why?
Is it? Because the capsules still? Or like they can still release toxins.
They can still hurt you. So it is false,
Yeah.
you're right. Okay. That one's too easy. Too easy. Oh, true or false? Human cells can't get hurt by lipozyme.
By what? Sorry?
Lysozyme. True or false? Human cells cannot get hurt by lysozyme. You know, like that's in the tears and then the mucous membrane.
So we don't have peptidoglycan?
Good job, so it would be true. Human cells can't. Blank is normally back and forth motion or vibration of molecules.
Oh yeah.
You're right, grounding motion. Very good. Okay, blank is a protein channel that helps water into a cell. Think Spanish. Yeah, I heard it.
Aquaporin.
Aquaporin. Very nice.
Aquaporin.
Okay. Blank diffusion is the movement from an area of high concentration to an area of low concentration.
High to low, is that what you said?
Uh-huh. Blank diffusion is the movement of a high area of concentration to a low area of concentration. Very good. Simple. It's simple diffusion.
Oh. That's what we meant.
Okay, blank diffusion transports material from a high concentration to an area of low concentration. Blank diffusion transports material from high to low.
Requires work, yeah. Active transport.
Nope.
No.
Facilitated?
Facilitated. Very good. Facilitated. Okay. Blank reactions transfer energy from a complex molecule to ATP. Think of your cat.
Catatonic.
Catabolic.
Catatonic.
Catatonic. That's awesome.
This is why I should
Catabolic. Okay.
It's Friday night.
It's awesome. Okay, 60% of ATP during the catabolic reaction turns into what? What?
Pyruvate.
Heat. Very nice.
What?
Heat.
What was it?
Turns into heat.
Oh. Turns into This Friday night.
Tools that are involved in making ATP are
Electron transport, is that what you mean? Is like
no, not
okay.
specific. I'm just asking for like a general tool that helps make ATP. Like maybe moves it along, speeds up the reaction.
Enzymes?
Oh,
Good job.
catalyst?
Enzymes.
Just kidding.
Catalyst, which is an enzyme.
Yeah, catalyst.
Which is an enzyme, yeah. It's all good. Blank line up the molecules so the chemical reactions happen more easily. Can we just talk about this?
Enzymes?
Yeah. Remember, like they go out and they grab it. You got this little enzymes and it grabs this one, and then they meet together. And they go together, so it makes it faster instead of just going around with the brownie in motion, bumping into everyone until they finally meet their match. This is like the dating, like I heart you or something, dating online. And so boy meets girl and there's love. Instead of just, fumbling around college trying to find your mate. You actually go to a dating site. I don't know. Let's see. Cofactors are the blank part of the enzyme plus the protein portion. A hopso enzyme make what?
Hollow enzyme.
The hollow enzyme, good job, but what are the cofactors?
I should have wrote it from chemistry. Does that have something to do with binding?
What? She said it? I didn't hear it.
They're ions.
Like metal ions?
It's the vitamin and mineral. Vitamin and mineral, the cofactors are the vitamin and mineral part of the enzyme. So, yeah, could be mineral. Oh, please don't show. Crap. Blank coenzymes pick up hydrogen ions, making them blank. That's taking the hydrogen ion down to the electron transport chain. Yes! Who said that? NDA! Woo! Go Logan! It's the NDA coenzymes pick up the hydrogen ions, making them what?
Is it?
Did I say NHD? Oh, I don't know what I said. It's NAD.
NAD, like, picks up the hydrogen.
Yeah, so what does that mean? What is it called? What?
I make some NADH.
That's what I'm looking for,
Oh.
NADH. They pick up the hydrogen ion, making them NADH. Sorry. Okay. Blank blank is essential for the metabolism in cells. Wow, I can't talk. It is Friday night. Blank blank is essential for metabolic reaction in cells. This one was a while ago. I'm trying to think of what we put for it. Sulfonylamides block this, I believe.
PABA?
Oh, the PABA?
PABA, but what are they blocking? Fluoric acid.
Something about the acid.
Folic acid.
Folic acid.
Folic acid.
Well,
Let's see.
Well-
Proteins can be blank by excessive heat, acidity, or by a strong base. Denatured. Very good. Okay.
nature.
Denatured. Oh, nerve gases typically cause blank, non-competitive, enzymatic inhibition of enzymes.
Wait, will you repeat that?
Nerve gas typically causes blank, non-competitive, now I can't say it, inhibition of enzymes. Enzymatic.
Reversible or irreversible?
Irreversible. Very good. Do you remember the competitive and inhibitory? Do you remember how it would come in? So you have this binding site, and you'd have the what am I trying to say? The compatible?
Competitive or? not?
Competitive, thank you. The competitive come in and will come in here, and it will bounce off the enzyme that's trying to attach. Does that make sense? So it's competing for it, kind of like a basketball game. This is reversible. It will come and go. But I think it's the allosteric site?
Allosteric.
Yeah, allosteric site.
Yeah.
Thank you. That's the on and off switch. That's where it can come on and come out. Then you have the one for nerve gas, which actually comes down here and binds right there. And it changes the binding site, so it can no longer bind to anything. So it's a permanent change. That's what nerve gas does. It's permanently changing. Does that make sense? Anyway, do you guys understand the cellular respiration? Do you want to go over that at all, or are you guys good? Good, bad, ugly?
Good.
Good.
No.
All right, we'll move on. Okay, next test. How much longer do we have?
How long are you staying for?
I think we got it broadcast until 7, but I'll stay as long as you guys want to if you're on.
Wait,
Yeah, of course. Go ahead. I'll get this up and going. I did hear you. Mom.
I'm not used to mics. I don't like this.
Oh, are you serious? What do these mean?
Neutral and dead.
Yeah, so stop using this.
Not with disease and without disease, or with, oh,
Very nice. It's the same thing. It's with disease or with contamination or without contamination. So you're right.
I have that picture in my mind, the little giant microbe he brought in, but it wasn't that one, it was on the website, it was like yellow, like all these little things going around it that were like wavy.
Honestly,
Okay.
I don't remember this one. Yeah, I don't remember that one on the test, which what are the hardest things to kill? Spores?
Endospores.
Endospores, prions.
Prions.
Prions are even harder to kill than endospores. They're the bigger part.
That's the one that causes mad cow. The prion.
Do you remember how that works? I thought it was cool.
It's a misfolded protein.
And it bumps into the other proteins, and they keep changing to look like the misfolded protein, so it just keeps
Oh.
going and going and going and
Almost
going.
like a cancer or something.
microbes on desiccated food?
Like, so drying food,
Uh-huh.
like drying grapes for raisins and such.
And jerky.
And then would be mold.
Very nice. Why? What kind of environment does mold like to live in?
Like,
Water,
well,
it's dry.
really dry,
Hypertonic.
hypertonic, that word.
Genotypes. What you are, phenotype is what you see.
The blueprint for builder. Very good. Ugh. Still wouldn't do it. This was brand new right out of the package. I wouldn't do it.
Really?
I don't know.
Disinfection, sterile, sterilization.
Sterilization.
Yeah.
That kills all of them. Disinfection only chose the active ones.
I just had to try it.
Oh, you would? Yeah, I kind of. Ugh. Okay. You're sanitizing, you're only sanding off part of them. Germicide to kill the germs of the microbes. Degerming is like hand washing. Antiseptics, I always think of Anna, so like a person, so it's the skin. What factors can affect the efficiency of a given method of microbial control?
Time.
That's good. What else? Mm-hmm.
Kind.
Do you remember the mean one? Oh, I guess it says it right there. The Pseudomonas argenteus. I can
Aruginous.
whatever. I'm almost positive that he'd have at least one question because he
He
kept.
always talks about that.
Yeah, and he kept bringing it back up and back up. So, what are some characteristics of this?
Mhm.
Isn't it the blue-green one?
Mm-hmm. What was the smell like?
Sweet?
It was sweet.
Really?
Yeah. This was the one that had the pump that if you put antibiotics to attack it, it would pump it right out.
And what kind of environment does it like?
Yeah, what kind of environment does it like? That's good.
You guys remember?
The human body likes.
Goes to more?
Moist, watery.
Moist and watery is the most.
So not necessarily warm.
Yeah.
Just moist.
Yeah. It likes to live in a lot in the hospitals all the time.
Yeah,
Oh.
likes to live on the kid.
It's very antibiotic resistant.
Your kid?
Yeah.
I'm sorry. You better now or?
He's on and off antibiotics, 30 days on, 30 days off for it. Has been for about 3 years.
What kind of symptoms does it give him?
It's, he's got a rod in his spine for scoliosis and it just grows on that. So it causes sores and stuff like that.
Oh.
Poor kid. Oh. Pretty strong. Wow. Holy crap.
It's strong stuff.
The diplomized, is that what you said?
Total nice.
Diplomized.
He gave that to my dad when he was in ICU.
Sorry.
When they didn't know what he had, but he went into endotoxic shock, and that's heavy stuff.
Yeah.
Dang.
Is it a broad spectrum or is it a?
I think it is.
I would
Triple
think so.
myosin, is that what you said?
Toby mice.
Oh, I keep getting it all mixed up.
Yeah. Anyway, Logan, what do you think? All right.
Like by what action? Oh, never mind.
It's all good. So they can pop the bubble or they will damage the DNA. That's usually what they are going after. What type of machine keeps anthrax from going through the mail?
UV light.
High energy electron transport beams. They form the ionizing radiation. They don't penetrate well, but they work well for anthrax through the little mold. What are the types of heating that are used to denature proteins and bacteria? Temperatures and times.
Huh?
For autoclaving, there's the 15, 121, 15, so it's for 15 minutes at 121 degrees Celsius at 15 pounds of pressure. And then there's pasteurization. I think you're thinking about dry heat. But pasteurization, there's 63 degrees that's for 30 minutes. Because then 72 is for 15 seconds.
Oh, right.
And then the dry heat is like, is it 170 for 2 hours?
Yeah. Good job, guys. I would know these. If I remember right, there was a test question. Hey, is that up?
What?
Is that up?
Yeah,
Oh,
it's
okay.
going. We're
How long
rolling.
has it been going?
8 minutes, 9 minutes.
Oh, really? Okay. What type of disinfectant would be best to kill the microbes in plastic? What about alcohol?
Probably. I don't know.
Fennel?
Oh,
Is alcohol good at cleaning out wounds?
no.
Why?
Because it makes them like just stop the top of your wound or something like that. And then they don't really leave or anything.
Alright. Salmonella has a generation time of 30 minutes. If 4 cells are present on a turkey that is left on the counter, how many cells will be present after 3 hours?
Oh wait.
One more.
Who's close enough? You guys want to go through that, or you got it? We're good. Okay.
Wait, can we go over that last one?
Okay. Okay. First of all, you got 30 minutes, and each time, since it's generation time, it's going to be doubling, right? So you got 30 minutes, and we're going for 3 hours, right? So there's 2 hours, there's 3 hours. All right. So you start with 4. 4 times 4 is what? 8? 8 times 8 is 16. 16 times 16 is, I don't know.
Close. You're adding.
Oh, yeah, you're adding.
Okay.
Sorry.
Just making sure.
64, 128, 256.
You know times when you add them. To me, it makes sense. Sorry. Does that make sense? Can you all see that? No, you can't. Can you see me? That's weird. Do it here. So you got 30. Okay, you don't have any.
I still don't think I can see. There you
Really?
Like right where you are, you should write it.
Right here?
Yeah.
All right, we'll try there. Can you see? You can't even see that. Well, this sucks.
They should be able to.
You're in the picture now.
But I can't even see it. Can you guys see that? And Logan? Anyway, all you're adding, what I like to do is make kind of a graph. I wonder if I can change it to. You in there, Logan? Logan? 3 hours, every 30 minutes. Okay, so you got 1 hour, 2 hour, 3 hours. So 4 plus 4 is 8. 8 plus 8 is 16. 16 plus 16 is 32. 32 plus 32 is 64. 64 plus 64 is 128. 128 plus 28 is 256. That make sense? Yes? I'm gonna see this. Does that make sense, Logan?
Bueno.